First question of this paper says that fungal cells and plant cells contain mitochondria. They then ask us to state the function of mitochondria. Mitochondria can be found in all eukaryotic cells, meaning that the only cells that don't have mitochondria are things like bacteria and then also archaea. Now, mitochondria is responsible for aerobic respiration, and the reason why I don't just say respiration overall is because the first part of respiration, which is the responsible for anaerobic respiration, meaning that it does not require oxygen, actually happens in the cytoplasm outside of the mitochondria. Only the second and the last part of respiration, which requires oxygen, occurs within the mitochondria. So the mitochondria will be responsible for aerobic respiration. Now it's important to remember that mitochondria are not the same as chloroplasts. Chloroplasts are responsible for producing carbohydrates from light energy, whereas mitochondria are responsible for producing energy or ATP from carbohydrates. So the products that are made from chloroplasts are used as the substrates in mitochondria for aerobic respiration. Now we can answer question A as aerobic respiration. Now question A2 says state one feature of plants that's used to distinguish them from fungi. So we can go and look at the different kingdoms that we get and look at the differences between them. We can first divide all living organisms into two main groups called prokaryotes and eukaryotes. Prokaryotes include organisms like bacteria. Bacteria are single-celled organisms and they consist of a cell wall made up of peptidoglycan. They have no nucleus. Instead of a nucleus, they have a circular piece of DNA. They also contain these smaller pieces of circular DNA called plasmids, which are very important for humans as we use them in biotechnology and genetic engineering. Prokaryotes, like bacteria, also have no membrane-bound organelles, meaning that they won't have things like chloroplasts and mitochondria. We then have our eukaryotes. Our eukaryotes include plants, animal, protists, and of course fungi. Both plants and fungi have a cell wall as well as a cell membrane. The cell wall in plants are made up of cellulose, whereas the cell wall in fungi is made up of chitin. Animals and protists only have a cell membrane. Now all of these eukaryotes have a definite nucleus that include the nuclear membrane with the nuclear plasm. All of them also have mitochondria for cellular respiration. Only plants and protists have chloroplasts as they are autotrophs and use sunlight to produce carbohydrates. Animals and fungi are known as heterotrophs and needs to find carbohydrates from consuming different types of food. One characteristic that sets protists apart is its contractile vacuole. This is because protists live in a watery environment and needs to be able to eject the water from the cell, otherwise they will burst. Plants have a large permanent vacuole and animals have tiny vacuoles. Some are called lysosomes. Then fungi can have several large or small vacuoles. So if we go back to our question, we can state that plants have cell walls made up of cellulose and fungi have cell walls made up of chitin. We can also say that plants have chloroplasts whereas fungi do not. The next question says that yeast is a fungus that can respire to produce ethanol. State the balanced chemical equation for this type of respiration in yeast. Before we answer this question, let's look at the different types of respiration and how they occur. The respiration can be divided into two main types. The first one is aerobic respiration that requires oxygen to occur. So this is where glucose plus oxygen will produce carbon dioxide and water. This usually occurs within the mitochondria. We then also get anaerobic respiration, which does not require oxygen. Now, this can occur in humans during intense exercise, and here glucose would be respirated into lactic acid. Now, lactic acid is that acid that sits in our muscles and makes us stiff the next day. This can also occur in yeast in the absence of oxygen. In yeast, the glucose is being respired into ethanol and carbon dioxide that can be used in biotechnology. So to answer this question, we can say C6H12O6, which is glucose, will produce 2 C2H5OH, which is ethanol, plus 2 CO2, which is carbon dioxide. The next question states, a scientist investigated the effect of sugar on respiration in yeast cells. One flask contained 100 centimeters cubed of a sugar solution, and another flask contained 100 centimeters cubed of water. Both flasks contained the same mass of yeast. The temperature was maintained at 25 degrees Celsius. The scientists used the apparatus shown in figure 1.1. So in this figure, we can see that they have a water bath at 25 degrees Celsius. This will keep the temperature constant. They then have an Erlenmeyer flask or a volumetric flask with yeast in a sugar solution covered by oil, a thermometer to check the temperature, and this all is connected to a syringe to collect the gas that's being produced. On the next page, they give us figure 1.2, which is a graph of the results of the investigation. So this graph shows the volume of carbon dioxide gas that's being produced in a certain amount of time for yeast in a sugar solution at 25 degrees Celsius, and then also for yeast in water at 25 degrees Celsius. Question 1 then asks, using the gradient shown in figure 1.2, calculate the rate of carbon dioxide gas produced by the yeast in a sugar solution between 10 minutes and 15 minutes. We also need to include the unit. Now, the rate will always be equal to the change in y over the change in x. In this example, my change in y is the volume of carbon dioxide gas produced, and my change in x will be the time in minutes. So my rate will be the centimeters cubed over minutes. So if I look at the change in y between 10 and 15 minutes, I can see at 10 minutes it's 11 centimeters cubed, and at 15 minutes it's 19 centimeters cubed. So I'll say 19 minus 11, this will give me 8, and this will be divided by my change in time, which is 15 minus 10, and this gives me 8 over 5, or 1.6 centimeters cubed per minute. They then asked to give a reason for the oil layer in the apparatus showed in figure 1.1. Now, the oil layer on the sucrose solution prevents oxygen from entering the sucrose and yeast suspension so that anaerobic respiration can take place. But carbon dioxide is still able to diffuse from the solution out through the oil suspension to be taken up by the syringe. On the next page, question 3 asks to state one reason why no more carbon dioxide gas was produced after 35 minutes by the yeast in the sugar solution. Let's have a look on how to interpret a rate graph. So we can have the amount of product formed over time to be able to get a rate graph. Now, as my graph increases, it means that the amount of product formed will increase. In this example, it increases pretty uniformly, meaning that something else is keeping the reaction from increasing even faster. This can be the amount of enzyme, the temperatures, or the amount of organisms in my solution. Now, the second part of my graph is where my graph plateaus. It means that no more product is being formed, that the amount of product being formed is now at a maximum. This means that there would be no substrate left for the reaction to continue. We can then answer the question as follows. All the glucose or substrate in the solution has been used up by the yeast cells during respiration. The next question says that the scientists repeated the investigation using yeast and a sugar solution at a temperature of 95 degrees Celsius. Explain why no product was formed during this reaction. Now, to understand how temperature affects a reaction, we have to go and have a look at enzymes. Now, if this is my enzymes are affected by temperature and pH. If I look at a graph of temperature over the activity of an enzyme, my graph will kind of look like this. So in phase 1, my temperature increases. As my temperature increases, my kinetic energy will also increase and particles will move faster, increasing my rate of reaction. 2, this is my optimum temperature, and this is the temperature by which my reaction will occur at its fastest. 3 will be where my enzyme denatures. I then have my pH. Now my pH graph is a lot more uniform. So phase 1, when the pH is too low or too high, is where my bonds will distort. And phase 2 is my optimum pH, where the rate of the reaction of the enzyme will be at its highest. Where the green stars are present, this is where my bonds in the active site will distort, meaning that the shape of the active site will slightly differ. This will make it more difficult for the substrate to bind to the active site. But this is reversible when we lower the temperature or we go back to the normal or optimum pH, then the active site will go back to the normal shape and the rate of reaction will continue. Now here with my orange star, where the enzyme denatures, this is when the bonds in the active site break, and this will mean that the active site will lose its shape entirely and the substrate will not fit into the active site at all. This process is irreversible. So we can answer this question by saying that respiration is catalyzed by enzymes. So at very high temperatures, the bonds at the active site will distort and eventually break, causing the enzyme to be denatured. The substrate will no longer be complementary to the active site and no products will be produced. If we look at the next question, question D, it asks to state one way in which humans use carbon dioxide gas produced by yeast cells. So to answer this question, we have to look at what we use the products of anaerobic respiration for. So we know that yeast undergoes anaerobic respiration, and in this reaction, glucose is converted to ethanol and carbon dioxide. Now the ethanol can be used in fermentation, like in beer making or kombucha making. The ethanol can also be distilled further to be used in more expensive wine making, vodka making, or any other type of alcohol products. Then we have carbon dioxide. So carbon dioxide is used as a leavening agent. This means that we can use it to make bread or pizza dough as it makes the dough rise. Now remember that the ethanol will evaporate at low temperatures and will not be incorporated into the bread. That is why we don't get drunk when we eat too much bread. So one way in which carbon dioxide can be used is to make bread rise, so as a leavening agent. The next question asks to state the name of one gas other than carbon dioxide that contributes to the enhanced greenhouse effect. So to be able to answer this, we need to go look at all the greenhouse gases. The Earth is covered in a natural ozone layer. Now this ozone layer lets short-wave radiation or incoming radiation from the sun through, but it stops some of the long-wave radiation from escaping. This helps to control the temperatures on Earth and makes it possible for all things living to thrive. Now, because of pollution, we actually add greenhouse gases to our ozone layer. This means that less radiation will be able to escape, and the Earth's temperatures will increase, leading to the ice caps melting, our ecosystems changing, and some species going extinct. Now, these greenhouse gases include gases like carbon dioxide, methane, CFCs or chlorofluorocarbons, and then nitrous oxide. So to answer this question, you can name any of those gases except carbon dioxide. So you can say methane, you can say nitrous oxide, you can say chlorofluorocarbons, or any other greenhouse gas that you know of. On the next page, they ask us a few questions about the nervous system. So let's quickly go through the reflex arc and the nervous system itself. Here we have a diagram representing the reflex arc. We have the skin, which has receptors inside of the skin just underneath the outer layer. We then have our sensory neuron, which is then connected to our relay or our interneuron, which is in turn connected to our motor neuron, which ends at the effector or muscle. The receptors in the skin will detect a stimulus and send an impulse through the sensory neuron towards the relay neuron. The relay neuron will then continue the stimulus to the motor neuron that would then continue the impulse to the effector muscle. The effector muscle will then respond towards the stimulus. Now here where the orange stars are my synapses. This is where one neuron connects to another neuron. The synapses consist of a presynaptic membrane, the synaptic gap, and then a postsynaptic membrane. In the presynaptic membrane, there's also mitochondria and vesicles that contain the neurotransmitters. On the postsynaptic membrane, there are little receptor molecules that are complementary to the neurotransmitters. So when an impulse approaches the presynaptic membrane, it will cause the little vesicles to fuse with the presynaptic membrane, releasing the neurotransmitters into the synaptic gap. The neurotransmitters will then diffuse across the synaptic gap and connect to the receptor molecules on the postsynaptic membrane. This will then induce an impulse to be continued in the next neuron. So, our first question asks us to define the term sense organ. Now, this would be a group of receptor cells that detect a stimuli in the external environment and then transmits an impulse to the sensory neuron. In question B, they give us figure 2.1, which shows us the structure of an eye. It also shows the pathway taken by the nerve impulse, which helps bring about the changes in the eye in order to bring light to focus. They then ask us to draw a label line and label it X on figure 2.1 to identify the motor neuron. Now, the motor neuron would be from the brain towards an effector cell. So it would be the top line. Then, in question 2, they're asked to describe how the events that occur at the synapse generate an impulse in the next neuron. So here we can say that neurotransmitters are released from vesicles that would then fuse with the presynaptic membrane. These neurotransmitter molecules will then diffuse across the synaptic gap and bind to receptor molecules on the membrane of the postsynaptic neuron. On the next page, we have the third question that asks to describe and explain the process of accommodation in the eye to view a near object. They also ask that we use the letters in figure 2.1 in our answer. After this, in C, they also ask us to describe the distribution and function of rods and cones in the eye. So before we answer these two questions, let's have a look at the eye and different processes that occur in the eye. So here we have a diagram showing the eye. The first part that we're going to look at is the optic disc. Now this is where the optic nerve meets the eye. Disc has no rods or cones or any type of photoreceptors. So when light falls on this part of the eye, it will not be transmitted. The next part that we have is of course the optic nerve, and this sends impulses to our brain to say what we see and how to respond. We then have our blood vessels that supply the eye with oxygen and takes away carbon dioxide. The next part of the eye that we have is in the cornea, which is a clear dome-shaped layer that covers the front of the eye, and it has several functions. So the first function is that it focuses light, and it also protects the eye. It shields the eye from dust and bacteria and other harmful particles, and it also shares this protective role with the eyelids, the tears, and the eye socket. Then we have the iris. Now the iris is the colored part of our eye, and it surrounds the pupil. It regulates the amount of light that enters the eye, and we will soon look at how it does this. We then have the pupil, which is the opening into the eye cavity. This allows light to enter the eye and fall onto the photoreceptors. We then have the lens in the eye, and the lens focuses light onto the retina, which is the light-sensitive tissue inside the eye. Now, for the lens to be able to focus the light, the suspensory ligaments and the ciliary body work together to change the shape of the lens to be able to bend the light. We then have the sclera. Now this is the white part of the eye, and it has many functions, including the protection of the eye. It also gives the eye its shape. It also provides support for the ciliary muscles. The next layer is the choroid, and it also has several functions. So the first one is light management. It makes it easier for the retinas to process the light they receive. It also regulates temperature and volume. It also releases growth hormones, which plays a role in the adjustment of the eye shape during growth. Then we have the retina. Now the retina's function is to convert light into electrical signals that the brain interprets as images. It has two types of photoreceptors called rods and cones. Now rods are responsible for detecting low light intensities and develops an image in black and white, whereas cones are able to interpret high light intensity and develops images in color. Lastly, we have the fovea, which is responsible to provide sharp central vision. This is essential for tasks that require visual detail, like reading or driving. Now the fovea only has cone photoreceptors and are able to make sharp color vision possible. So the lens of our eye is able to change shape so that we are able to focus on either distant or near objects. This is called accommodation. So let's look at distant objects. When the light rays come into the eye, they get concentrated on the pupil and the lens. The ciliary body or the ciliary muscle will then relax. This means that the suspensory ligaments will tighten, forcing the lens to become thinner. This will force the light rays onto the fovea, giving us a clear image. We then have near objects. So as the light enter the eye, it will be focused on the pupil and the lens again. The ciliary muscle will then contract, lengthening the suspensory ligaments, which will then make the lens flatter and thicker. This will then bend the light rays to be focused on the fovea again. The eye is also able to control the amount of light that enters the eye, and it does this through the use of two types of antagonistic muscles. Now, the first of these is the radial muscles, and the second of these is the circular muscles. Now, the radial muscles are from the pupil to the edge of the eye, and the circular muscles move in a circle around the eye. Now, when we are in dim light or when there's not a lot of light available, the pupil wants to enlarge to allow more light to enter. So our radial muscles will contract and shorten, whereas our circular muscles will relax and enlarge to open up the space of the pupil. When we are in a situation that has bright light or a lot of light, our pupil wants to become smaller to allow less light to enter the eye. This will mean that our radial muscles will relax and become longer, and our circular muscles will contract and become shorter. Let's quickly go back to the previous page to the diagram of the eye that they gave us and name the different parts. So A would be the retina, B the fovea, C will be the blind spot, D is our optic nerve, E our ciliary muscle, F our lens, G our cornea, H our conjunctiva, J our iris, and K our suspensory muscle. So, the first question that asks to describe and explain the process of accommodation in the eye to view a near object, we can answer in the following way. We can say that the ciliary muscle E will contract. The suspensory ligament K will loosen. This will cause the lens, which is F, to become more convex, and this will cause more refraction to occur and light will be focused on the fovea. Question C asks to describe the distribution and function of rods and cones in the eye. So we now know that both rods and cones are found in the retina. There are no rods found in the fovea, only cones, and the blind spot has no rods or cones. Now both rods and cones are light receptors that are able to detect light and send an impulse to the sensory neuron. Rods are able to detect low light intensity, whereas cones are able to detect high light intensity and produce color vision. On the next page, we have question D. It says that red-green color blindness is a sex-linked characteristic. It is controlled by a gene on the X chromosome. There are two alternative versions of this gene: no color blindness, which is X with a capital A, and color blindness, which is X with a lowercase a. So before we answer the questions, let's go look at sex-linked characteristics. Sex-linked characteristics is when a gene with its alleles are located on one of our X or Y chromosomes, meaning our sex chromosomes. Now we need to remember that males have two X chromosomes and females have an X and a Y chromosome. Now the X chromosome always comes from your mother, whereas the Y chromosome always comes from your father. Now the X chromosome is a normal sized chromosome and is able to carry a lot of different genes. The Y chromosome, being much smaller than the X chromosome, lacks some of the potential genes. Now, if we look at the female sex chromosomes, we have an X and an X, meaning that we will be able to be heterozygous for a gene on our sex chromosomes. One can be dominant, one can be recessive. If the recessive gene is the disease, the dominant gene, the capital letter, will mask the trait of the disease. But if we look at males that have an X and a Y chromosome, and only the X is able to carry the gene, if they have one version of the recessive diseased gene, there's no backup X chromosome that's able to mask the trait. Okay, now let's look at the question. Question 1 says, state the term used to describe an alternate version of a gene. Now this is called an allele. We get dominant alleles and recessive alleles. They then ask us to state the genotype of a male with color blindness. Now we know color blindness is a recessive gene. So for a male to have color blindness, it needs to have the recessive gene on its X chromosome. So it will be X with a recessive B and a Y. On the next page, we have question 3. Now, A says meiosis and mitosis are important processes in life cycles of organisms. Figure 3.1 shows the life cycles of two different organisms. organism 1 has a simple life cycle and organism 2 has a complex life cycle. It has a stage A that produces spores and a stage B that produces gametes. In the diagrams, the haploid number of chromosomes are represented by N and the diploid number of chromosomes are represented by 2N. If we look at organism 1 and we start at the adult phase, we can see the adult phase being 2N then breaks up into two separate phases called sperm and egg. These two then come together to form a zygote, which then produces an embryo. So we can already see that this is kind of the life cycle like a human or any other mammal's life cycle. We then have organism 2. So let's look at the multicellular stage A, which is basically the adult stage, as 2N. It will then form spores, so it goes from 2N to N, and then a multicellular stage, which is only N, which then splits into female gametes and male gametes that forms a zygote and then forms an embryo. This looks similar to the life cycle of other fungi. So on the next page, we have question 1. So question 1 says table 3.1 shows the letters P to T in figure 3.1 and the type of nuclear division. We need to place a tick in each row to indicate whether it's mitosis or meiosis. So let's quickly look at the difference between mitosis and meiosis so that we can identify the different phases. Mitosis and meiosis both form part of the cell cycle. The cell cycle can be divided into 4 distinct phases. The first one is the G1 phase, with G standing for growth. So this is where the cell grows and increases its volume. We then have the S phase. Now S stands for synthesis. This is where it duplicates its DNA. Then we have the G2 phase, and this is the second growth phase. So again, increase in size. And then lastly, we have the M phase, which is either mitosis or meiosis. Let's firstly look at mitosis in the cell cycle. So the first stage of mitosis is the prophase. Here is where the chromatin condenses into chromosomes. The nuclear envelope will break down and the centromeres would move to opposite sides of the cell. The next phase is metaphase. Here the chromosomes will condense more and move together, aligning on the center of the cell. This is called the metaphase plate. We then have anaphase, which is where the physical connection between the sister chromosomes are severed and the sister chromosomes will be separated to opposite ends of the cell. Then we have telophase. This is where the mitotic spindle breaks down, the chromosomes will start uncoiling and the nuclear membrane will reform. Lastly, we have cytokinesis. This is where invagination of the cytoplasm will occur and cause the cell to split into 2 different cells. Now, the important thing about mitosis is that it starts with double the chromosome number, so 4N, and then it ends with the normal amount of chromosomes, which in this case will be 2N, which means that we end up with 2 genetically identical cells. I say genetically, that's because some things in the cell might not be identical, like the amount of cytoplasm. Mitosis occurs in our stem cells in our body and it's responsible for the growth of beings, asexual reproduction like in bacteria, and then also the replacement of cells. We can now have a look at meiosis. Now, meiosis has similar phases as mitosis. It starts off with prophase 1. Just as in mitosis, in prophase 1, the chromatin condenses into chromosomes. Here we have double the chromosome number. We then go to metaphase 1. Just as in mitosis, the chromosomes align on the metaphase plate, but what is different is that they pair in their homologous pairs. The separate chromosomes, not just the chromatids, are then separated in anaphase to opposite poles. During telophase 1, the homologous chromosomes is now separated. They then reform the nuclear envelope around each set of chromosomes, and then the cell kind of goes into a resting phase instead of cytokinesis. We then move on to prophase 2. So in prophase 2, the chromatin will recondense into chromosomes. We will then move to metaphase 2. Just as in mitosis, in metaphase 2, the chromatids are then aligned across the metaphase plate. The spindle fibers will attach to the centromere and pull these chromatids to opposite ends of each cell in anaphase 2. We then go to telophase 2. These are when the chromosomes decondense into chromatids. The nuclear envelopes reform around the chromosomes and they go through cytokinesis that would then split into 4 separate cells. So in meiosis, our final chromosome number in our 4 cells is N and not 2N such as in mitosis. This means that we get 4 genetically different cells from each other. Now this is very important as it gives us more diversity and more genetic diversity when it comes to reproduction. This is why you and your siblings don't look alike at all. Now, meiosis is only used in the production of gametes such as sperm cells, egg cells, and pollen cells. We can now go back to our diagram and go identify the different phases. So the first one that we can identify is P. So if we look at P, we see an adult with 2N that is separated into sperm and egg. This means that this would be part of meiosis as gametes are formed. We can then look at Q. Now Q goes from an embryo to an adult, meaning that it's just the growth of the organism. This will be mitosis. We then have R. Now R goes from 2N to N, which is the production of gametes again, i.e. meiosis. And then S is a multicellular stage N that turns into male gametes, also N. This is a form of asexual reproduction, so it will be an example of mitosis. Lastly, we have T, where zygote becomes an embryo. This is growth, meaning that would be part of mitosis. Let's answer the question. So P will be meiosis, Q will be mitosis, R would be meiosis again, and S mitosis, and T mitosis as well. The next question asks us to identify and state the name of process W shown in figure 3.1. If we look at figure 3.1, process W is where the sperm and the egg fuses to form a zygote. This means that sperm, which has a chromosome number of N, and an egg cell, which has a chromosome number of N, will fuse together to form the zygote with a chromosome number of 2N. So if we had to answer this question, we can say that it is a form of fertilization. The next question says an embryo contains stem cells. Complete the sentences about stem cells and body cells. Now, stem cells are undifferentiated cells that divide by mitosis to produce daughter cells that can become differentiated for a specific function. Most body cells in an organism contains the same genes, but many genes in a particular cell are not expressed because the cell only makes the specific proteins that it needs. The next question asks to state the events in the life cycle diagram for organism 1 in figure 3.1 that would not be present in a life cycle diagram for asexual reproduction. Now, in asexual reproduction, the formation of gametes do not occur, and the division of the chromosomes do not occur. So we can say P, which is meiosis, will not occur, and we can also say W, which is fertilization, will not occur. On the next page, we have question 4. Here they say some algae are single-celled organisms that can photosynthesize. Their cells contain the pigment chlorophyll. State the name of one mineral ion that is needed to make chlorophyll. This is called magnesium. Now, a student investigated the effect of light on photosynthesis in algae using a hydrogen carbonate indicator solution. Now, carbon dioxide is an acidic gas. Table 4.1 shows the color of the hydrogen carbonate indicator at different pH values. So it shows at 7.6, which is more neutral, it would be yellow. At 8.4, which is a bit basic, red. And 9.2, which is more basic, it's purple. They say that 2 test tubes were set up at the same time. Both contained algae and hydrogen carbonate indicator. At the start of the investigation, the hydrogen carbonate indicator was red in both of the test tubes. One test tube was placed in the dark and one test tube was placed in the light. After 20 minutes, the contents of the test tube in the dark were yellow and the contents of the test tube in the light were purple. They then asked to explain these results. So let's look at what they did exactly. So they had 2 test tubes. At the start of the investigation, we can see that the hydrogen carbonate indicator is red. They then added single-celled algae to both test tubes. One was placed in light and the other one was placed in dark, so maybe like a container that keeps the light out. So in test tube 1, we can see that because it's in light, photosynthesis will occur. This means that carbon dioxide will be taken up with water and glucose and oxygen will be produced. Now, because carbon dioxide is used, it means it's making the acid less, so making the solution more basic. This means that the solution will turn a more purple color. Now in test tube 2, because it was placed in the dark, photosynthesis will not be able to occur because photosynthesis needs light. But respiration will still be able to occur. Now respiration is the process where oxygen is taken up with glucose and carbon dioxide and water is produced. Now, because carbon dioxide is produced, it means more acid is produced and it will decrease the pH. This means that the solution will turn a more yellow color. So we can explain the results by saying that algae will be able to photosynthesize in the light, but not in the dark, as light is needed to convert the carbon dioxide and water into carbohydrates. So in the test tube that was left in the light, the algae will absorb carbon dioxide. Because this is an acidic gas, it is being used up. The solution will become more alkaline and the indicator will become purple. In the dark test tube, the algae will respirate and release more carbon dioxide, making the solution more acidic and turn the indicator yellow. On the next page, we have the second part of the question, which says that figure 4.1 is a graph showing the effect of light intensity on the rate of photosynthesis. They then ask us to state which factors could be limiting the rate of photosynthesis at X and at Y. So if we look at X, we can see as light intensity increases, the rate of photosynthesis increases. This means that the limiting factor would be light intensity because we aren't giving it enough light intensity the whole time. That's why it increases as we give it a bit more each time. Then at Y, as the light intensity increases, the rate of photosynthesis stays the same, showing that even if we add more light, there's no increase in the rate of photosynthesis. It doesn't matter if we add double the amount of light or even triple the amount of light, meaning that something else must be limiting the rate of photosynthesis. This could be temperature or the carbon dioxide that's needed, or water, or even the number of chloroplasts in the plant. We then have question C. Now, question C says that starch and sucrose are made by plants after photosynthesis. State the name of the tissue that transports sucrose around the plant. So this is phloem, and the name of one other biological molecule that is transported in this structure, and that would be amino acids. Question C2 then asks to describe how starch is broken down in the human digestive system so that it can be absorbed by the blood. So let's quickly look at our digestive system and how it would digest a burger. So a burger is made up of buns that have starch in it, a patty with protein, maybe like a piece of cheese with lipids, a tomato that has vitamins, and some lettuce that contains water. So digestion will start in the mouth with mechanical digestion. This is our teeth grinding and chewing our food into tiny small pieces for us to be able to swallow it. Another digestion that happens in the mouth is chemical digestion, specifically amylase, which is an enzyme that digests starch. It digests starch into maltose. Lipase also works in the mouth and it starts digesting lipids. Food will then move down the esophagus into the stomach. So here mechanical digestion occurs again, and that is the churning of the stomach. This also breaks down the food into smaller pieces. Inside our stomach we also have pepsin, an enzyme that breaks down proteins into amino acids. Food then moves into our small intestine, and this is where bile will emulsify the fats. That is to break them down into small, tiny droplets of fats. In the small intestine, amylase also breaks down maltose into glucose. Here we can also find trypsin, which will break down proteins into amino acids as well. Now the difference between pepsin and trypsin is just their optimum pH. Pepsin has a much lower pH than trypsin. We also have lipase in our small intestine, and that will further break down fat. In our small intestines, vitamins and water will also be absorbed into the bloodstream. Our food will then move through our large intestine, where a small bit of water will be absorbed as well, and then out through our anus. So, if we had to describe the digestion of starch, we can say that it is broken down by physical and chemical digestion. So physical digestion will include chewing by the teeth in the mouth as well as churning in the stomach. This will increase the surface area of the food particles, which would increase the rate at which the chemical digestion can occur. So chemical digestion occurs where insoluble large molecules are broken down into smaller soluble molecules by enzymes. For starch, this starts in the mouth, where salivary glands will secrete amylase, which will break down starch into maltose. Maltose will further be broken down into glucose by maltase in the small intestine. On the next page, we have question 5. It says that figure 5.1 is a photograph of a koala. Koalas are marsupial mammals that give birth to offspring that are incompletely developed. The offspring develop further for several months inside the pouch of the mother. They're then asked to state one feature visible in figure 5.1 that could be used to identify the koalas as a mammal. So these features will be fur, and then also external ears, that is called pinna. Question B says that mammals use sexual reproduction to produce offspring. They then ask us to discuss the advantages of sexual reproduction in organisms such as the koala. So to answer this question, let's look at the advantages as well as the disadvantages between sexual reproduction and asexual reproduction. Now, sexual reproduction increases the genetic variation. It also means that species will be more adaptable. Diseases will be less likely to affect the whole population. Advantages of asexual reproduction include rapid growth. It can exploit a suitable environment, meaning that if the environment is suitable for one of the organisms, it will be suitable for all organisms. You don't require a mate, and it is very energy efficient. Now, the disadvantages of sexual reproduction includes that it takes time and energy, and a mate is required. The disadvantages of asexual reproduction include that there's limited genetic variation. The organisms will be vulnerable to habitat changes, and diseases are likely to affect the whole population. Evolution is also very slow in asexual reproduction. So we can see here that the advantages for sexual reproduction corresponds to the disadvantages of asexual reproduction. So, in summary, we can say that the advantages of sexual reproduction is that sexual reproduction allows for greater genetic variation, where different variations of alleles can be expressed. Thus, if an environmental condition changes, natural selection will occur, but because of a greater diversity of phenotypes, the offspring of sexual reproduction will have a greater chance of survival. Next, we have question C. So question C gives us figure 5.2, which is a diagram of part of the human female reproductive system, viewed from the side of the body. They then ask us to complete table 5.1 by writing the letters that identify the structures in figure 5.2, the names of the structures, and the functions. So let's quickly look at the male and the female reproductive system to be able to answer this question. Let's first have a look at the male reproductive system. So the first organ we have is the scrotum. Now the scrotum is there to protect the testes and regulate the temperature of the testes. Now the testes are responsible for making sperm and also involved in producing a hormone called testosterone. We then have the epididymis. Now the epididymis is the male sex accessory duct. So the main function of it is to store the sperm so that the sperm can mature and then be transported via the vas deferens to the urethra. We then have the seminal vesicle. Now the seminal vesicle is responsible for producing a fluid that makes a large portion of semen. It also helps to protect the sperm in the female reproductive tract, as it would make the fluid alkaline. We then have the prostate. Now the prostate is the actual organ that produces this seminal fluid that will be secreted by the seminal vesicle. We then have the urethra, which transports sperm, but it can also transport urine out of the male body. And then, of course, we have the penis. Now, the penis is responsible for two main things. The one is to become erect to be able to perform sexual reproduction, and the other one is for urination. We can then have a look at the female reproductive organ. So the first one is the ovary. Now the ovary is one of a pair of female glands that produces eggs and also the female hormone estrogen. We then have the oviduct, or also known as the fallopian tube. This is where fertilization takes place, and it carries the egg from the ovary to the uterus. Now the uterus is a pear-shaped organ inside the reproductive system. This is where the fetus will grow and develop. We then have the uterine lining, which is basically also called the endometrial lining and is responsible for the implantation of the egg cell. We then have our cervix. Now the cervix is a passageway from the vagina to the uterus. It also dilates when giving birth. And then, of course, we have the vagina, or the vaginal opening, which is the passageway from the outside world to the uterus. The first one that asks us to identify is the cervix. So we can see that the cervix would be M. Now the function of the cervix is to dilate when giving birth. The next one that we have to identify is the ovary. Now the ovary will be L, and the function of the ovary is to produce or release estrogen as well as an egg cell. We then have to identify the site of fertilization. Now this will be J, or the oviduct, and then the site of implantation, which would be P, or the uterus lining. The question then goes on to say that pregnant human females have a placenta. They're asked to describe the function of the placenta in humans. They then also say that the placenta is connected to the amniotic sac, and they ask 2 functions of the amniotic sac. So let's quickly look at a developing fetus inside the womb and the functions of each part. Firstly, we have the vaginal canal, and like previously stated, this is where sperm will enter the uterus, but it will also be the exit way for the fetus during birth. We then have the amniotic sac. Now, the primary function of the amniotic sac is to serve as a layer of protection for the developing fetus, but it has other functions as well, including the fact that it's a barrier from infection, and it also acts along the amniotic fluid to regulate the fetal body temperature. We then have the umbilical cord as well as the placenta and the placental villi. Now, these organs allow oxygen and nutrients to flow from the mother to the fetus, and then also waste material like carbon dioxide and urea from the fetus to the mother. So the blood of the mother and the blood of the fetus do not mix at all during the gestational period. Rather, the placental villi comes into really close contact with villi from the mother, so that the gas exchange can happen through diffusion. Lastly, we have the uterine wall. The uterine wall will be responsible for contractions during labor. Okay, let's answer the questions. So the first question asks us about the function of the placenta. So the placenta is responsible for the transfer of oxygen, nutrients, and antibodies from the mother to the fetus. It is also responsible for the transfer of carbon dioxide and excretory products from the fetus to the mother. So the placenta separates the maternal and the fetal blood, and thus any exchange of molecules takes place via diffusion. Lastly, the placenta is also responsible for the production and the excretion of some hormones. Now, the second question asks us about 2 functions of the amniotic fluid. Now, firstly, it provides support to the fetus. It also protects the fetus from mechanical shock. It maintains the temperature of the fetus, and it allows movement of the fetus. We could also say that it maintains the osmotic balance. It also helps the development of the digestive tract, and it provides a sterile environment for the fetus. Question 6 then asks to complete the sentences about the human teeth. So let's look at our human teeth. So the 4 front flat-shaped teeth are called our incisors. Next to them, we have 2 canines. These are made to bite and rip food apart. After that, we have 4 premolars. Premolars are there to help with chewing and grinding. Then at the back of our mouth, we have our molars. These are also flat, dull teeth that help with grinding of food. So we can answer this question by saying that the process of taking food into the mouth is called ingestion. The flat, sharp-edged teeth at the front of the mouth is called incisors and are used for biting off pieces of food. The premolars and the molars are the large teeth towards the back of the mouth and are used for chewing or grinding the food. They then give us figure 6.1, which is a diagram of a villus. On the next page, they ask us a few questions on the villi. So let's quickly go back to the previous page and look at the villi again. So, the villi is responsible for absorption in the small intestine. It is covered with microvilli, which increases the surface area of the villus. This means that nutrients can diffuse quicker across the villus. Underneath the microvilli, we have our capillaries. Our capillaries is responsible for absorbing several different nutrients as well as water. In the center, we have our lacteal. Now, our lacteal is responsible for the absorptions of glycerol and fatty acids. Let's have a look at those questions. So the first one asks us to draw labels and a label line on figure 6.1 to identify a capillary and a lacteal. So we now know where the capillary will be, just underneath the microvilli, and the lacteal would be in the center of the villus. Question 2 asks to state the part of the digestive system where villi are located. So they are located in the small intestine. Now question 2 asks to state the function of the lacteal. Now the lacteal is responsible for the absorption of fatty acids. The next question, question C, asks to describe the pathway taken by the products of protein digestion from the villi to the liver. So, remember, proteins are digested to amino acids. So the amino acids will move into the capillary and will be carried in the blood plasma to the hepatic portal vein.